Hey everyone, and welcome to this explainer. Have you ever wondered if we zoomed way in on a single drop of pond water, or maybe even explored a totally alien environment, how would we actually know if what we're looking at is alive? Well, today we're building the ultimate field guide to answer biology's absolute biggest question. What exactly separates the living from the non-living?
Okay, let's dive into this. Here's our agenda. We'll start off with the rules of life, then look at fueling the machine, compare eukaryotes and prokaryotes, meet the pathogens, and finally, we'll examine the ultimate biological rule breakers, viruses.
All right, section 1, the rules of life. So the acronym you really need to remember here is MRSCG-REN. You can kind of think of this as our ultimate universal cosmic checklist for identifying a living organism. It stands for movement, respiration, sensitivity, control, growth, reproduction, excretion, and nutrition. And here is the absolutely vital part to remember. If something lacks even one of these 8 capabilities, well, it isn't technically alive. It's an all-or-nothing biological requirement, no exceptions. Moving right along to section 2, fueling the machine. Look at the clear visual split in how organisms actually fulfill that nutrition rule we just talked about. On the left side, we've got producers, like plants. They harness sunlight, carbon dioxide, and water to literally synthesize their own food through photosynthesis. Pretty amazing, right? Then, over on the right, we have consumers, like animals and us. We are forced to eat other living organisms to get the complex energy and nutrients we need just to survive and grow. Now, once those nutrients are actually inside the organism, they have to be converted into usable power. Notice the formula for aerobic respiration here. And actually, a quick side note, don't confuse this with simply breathing. Respiration is a deep, cellular metabolic engine. It turns glucose and oxygen into ATP energy, and then releases carbon dioxide and water as a byproduct. And whenever a machine runs, it produces exhaust. In biology, that brings us to excretion. The metabolic reactions inside living cells produce waste, and honestly, some of this waste is highly toxic. In fact, right now, literally as you watch this explainer, your own body is actively carrying out this crucial role of life. You're continuously eliminating toxic byproducts, like carbon dioxide from your lungs and urea resulting from protein breakdown, just to stay alive. Talk about multitasking.
Okay, section 3, eukaryotes versus prokaryotes.
Let's look at the first group, the eukaryotes, which is just brilliantly diverse. Whether it's a microscopic protoctist, a branching fungus, a simple blade of grass, or an animal like that starfish right there, they are all connected by a shared, highly complex cellular structure.
But how exactly do they differ from prokaryotes? Well, while both groups share the basics, like having a cell membrane and cytoplasm, I want you to focus on the crucial point right in the middle. Prokaryotes completely lack a distinct membrane-bound nucleus, so their genetic material isn't locked safely in a vault. It's kind of just sitting right out there in the cytoplasm. Plus, prokaryotes are always single-celled and generally much smaller.
Let's crack open a prime example of a prokaryote, a bacterial cell. As you can see, you've got a peptidoglycan cell wall, a membrane, and cytoplasm. But look right at the center. There's no nucleus. It's just a single circular loop of DNA, often accompanied by these tiny extra rings we call plasmids. It completely lacks mitochondria and any other membrane-bound organelles. It really is a stripped-down, highly efficient, microscopic machine. And because they are so incredibly streamlined, single-celled bacteria can replicate themselves with terrifying speed through a process called asexual reproduction. It's a highly efficient step-by-step process. Step 1, the internal cell contents double. Step 2, cell division occurs as the cell starts to split. And finally, step 3, 2 genetically identical daughter cells are fully formed. You can really see that flow from top to bottom right here. The parental bacterial cell starts out by duplicating its circular chromosome. Then it literally pinches inward, splitting its doubled contents right down the middle to clone itself. Under the right conditions, this rapid replication can lead to massive population booms in a remarkably short amount of time.
All right, let's turn to section 4, meet the pathogens.
Now, the really scary thing about pathogens is that they aren't just confined to one single branch of our biological tree. Pathogens actually span across multiple groups. So you have pathogenic bacteria, like pneumococcus, which is a spherical bacterium that causes pneumonia. But then you also have pathogenic fungi, like certain species of mucor. And you even have pathogenic prototists, like Plasmodium falciparum, a microscopic parasite spread by mosquitoes that causes severe malaria in humans. They're basically everywhere.
Finally, Section 5, viruses, the ultimate rule breakers.
So the crucial point about viruses is totally evident right here when we look at their structure. You see an envelope, a protein coat, and a core of DNA or RNA. But what's missing? Absolutely everything else. There is no cytoplasm, no membrane, no ribosomes. Because they lack this basic cellular structure, viruses completely fail our initial MRSCGRN checklist. They don't respire, they don't grow, and they don't excrete. Since they're not truly alive, viruses literally cannot reproduce on their own. Instead, they act as obligate parasitic agents. To replicate, they have to physically invade a living host cell and aggressively hijack its internal metabolic pathways. They basically force the host's own cellular machinery to build multiple copies of the virus. So they're just mimicking reproduction by taking over the living. It's pretty wild. And these rule breakers indiscriminately target literally every type of living organism on Earth. Just briefly, let's look at 3 examples here. The tobacco mosaic virus infects agricultural crops like tobacco plants, destroying their chloroplasts and stopping photosynthesis dead in its tracks. Then there's HIV, a virus that ruthlessly targets and hides deep within the human immune system. And of course, the influenza virus, which attacks the cells lining our respiratory airways to cause the flu. So from plants to human immune cells, viruses really are a universal threat.
Which leaves us with this final, really provocative question. If viruses aren't technically alive, if they are essentially just undead packages of genetic code wandering around the microscopic world, how in the world are they so remarkably good at surviving, adapting, and dominating our biosphere? It's a mysterious paradox that challenges absolutely everything we think we know about life. So what do you think? I hope this sparked some serious curiosity for you today. Thanks so much for tuning into this explainer, and keep exploring the incredible world around us.